Welcome to Roadmap to Residency, Evidence-Based Applications. In this video, we will be talking about different bacterial cell components, and we will also be solving a few questions. So bacteria are the single-cell organisms, and cell wall is their key component. The cell wall protects the organism from the general support, osmotic pressure, target for immune system, and also it is a target for antibiotics. Now here you can see now this is the gram-positive. So the organisms that you can see in purple here are gram-positive, whereas those which you can see in pink are gram-negative organisms. So we'll talk in detail about this gram staining in further videos. So first of all, let's talk about peptidoglycan. So peptidoglycan is a major structural component of bacterial cell walls. It consists of polymer sheets of sugars and peptides. The sheets are cross-linked to other sheets, and the sugars that they contain are N-acetylglucosamine and N-acetylmuramic acid, so NAG and NAM molecules. Okay, the peptides are attached to the N-acetylmuramic acid, and there are 3 to 5 amino acids. The sugar peptide backbone makes the chain, and the chains are cross-linked by the peptide cross bridges. Site of action of some antibiotics is the peptidoglycan, like penicillin and cephalosporins. So peptidoglycan, so in gram-positive bacteria, there are up to 40 seeds, and 50% or more of the cell wall is made up of peptidoglycan. And in case of gram-negative bacteria, there are very few seeds, and only 5 to 10% of the cell wall is made up of peptidoglycan. So thick layer in the gram-positive organism retains the gram stain, which makes them purple. So there are some unique cell walls to some unique organisms. For example, in case of mycoplasma, they do not contain cell wall, and so they do not gram stain at all. The cell membrane has sterols for extra stability. In case of mycobacteria, the cell wall has mycolic acid. There is not gram stain well, and so we need to stain them with special stains like gel knee cell stain. In case of chlamydia, they lack the muramic acid. So cell membrane is present in both gram-positive and gram-negative bacteria. It is a phospholipid bilayer, and it helps in electron transport and oxidative phosphorylation. It consists of some enzymes as well as some carrier molecules needed for electron transport as well as oxidative phosphorylation. So the gram-positive bacteria, the unique feature is lipoteichoic acid. The major surface antigen for immune reaction is lipoteichoic acid. In animal studies, lipoteichoic acid has induced arthritis, uveitis, meningeal inflammation, cascades resulting in septic shock and multi-organ failure, induces cytokine release, and it binds the antibodies and it activates the complement cascade. Gram-negative, the unique feature is number one, periplasm. So periplasm, so the space between the cell membrane and outer membrane is called as periplasm, and they contain many enzymes. The most important among them is the beta-lactamase enzyme. Now, this beta-lactamase inactivates the antibiotics, so that you have some antibiotics which are beta-lactamase inhibitors. The unique feature number two is the outer membrane. Now, the outer membrane consists of lipopolysaccharide, which is a major immune trigger for gram-negative bacteria. And this lipopolysaccharide is what triggers the sepsis. Lipopolysaccharide contains of polysaccharide, lipid A, and O antigen. The lipid A is highly toxic and triggers cytokine release, and O antigen is the target for antibodies. So now let's talk about capsules. Capsules are the sticky gelatinous layer which are secreted by bacteria, helps attach to host cell, and protects against phagocytosis. Mostly water with some polysaccharide. So some special exception is Bacillus anthracis, where the capsule is made up of protein, and it is a major virulence factor, and it allows unimpeded growth for the organism. Talking about glycocalyx. So glycocalyx is a sugar coat made up of polysaccharides. It is similar to capsule, but bacteria with distinct, firmly attached gelatinous layer have a capsule. Bacteria with irregularly slimy fuzz layer have a glycocalyx. Understand the difference. So it is used to adhere to surfaces, that is catheters. For example, in case of a Staph epidermidis, which can cause biofilms. Okay. Now let's talk about Pili and Fimbriae. So Pili and Fimbriae are structurally similar to Flagella. It is made up of proteins and it has appendages or arm of bacteria. It allows the adherence to surface in case of ordinary Pili, and it attaches to another bacteria for conjugation in case of sex Pili. So the key bacteria are E. coli, which can cause UTIs or pyelonephritis. They contain the Pili and Fimbriae. The Neisseria gonorrhoeae, which shows antigenic variation by the Pili and Fimbriae. Now, this antigenic variation part is very, very important in case of Neisseria and gonorrhea. Please remember this part. Talking about plasmids. So plasmids are the small DNA molecule within a cell that physically separated from chromosomal DNA, can replicate independently, and can contain genes for antibiotic resistance. Toxins can be transferred from one bacteria to another. Let's talk about spores. Now, some bacteria can enter a dormant state called as spore, and they are then called as the spore-forming bacteria. They can survive for a long period of starvation, and these spores are resistant to dehydration, heat, and chemicals, but they do not have any sort of metabolic activity. Spore consist of coat, cortex, and dipicolinic acid. So the coat is the outermost layer, which is keratin-like protein, and it is impermeable to many chemicals like antibacterial agents. The cortex or core wall is the innermost layer, and it consists of peptidoglycans. And dipicolinic acid are in large amounts inside the spore, and they help with heat resistance. These are some of the spore-forming bacteria like Bacillus anthracis, Bacillus cereus, Clostridium perfringens, Clostridium britannii, or Clostridium botulinum. Here we have one question with us. So, which of the following virulence factors is most important for the development of this patient's condition? A 34-year-old woman comes to the office due to fever, dysuria, and flank pain. Temperature is 37.8 degrees Celsius. Blood pressure is 122 upon 78, and pulse is 84. Physical examination elicits suprapubic and costovertebral angle tenderness. Urine dipstick analysis reveals positive leukocyte esterase and nitrite. A subsequent urine culture shows colonies of motile gram-negative rods demonstrating a green metallic stain on eosine methylene blue agar and hemolysis and blood agar. Which of the following virulence factors is most important for the development of this patient's condition? Capsule, heat labile enterotoxin, lipid A, O antigen, or P. Fimbriae? This patient with fever, dysuria, and flank pain has bacterial growth in the urine. Findings diagnostic of urinary tract infection. E. coli, a motile gram-negative bacillus, is the most common cause of UTI and grows well on blood, McConkey, and eosine methylene blue agar plates. Now, due to its ability to ferment lactose, E. coli binds to dye in the EMB agar and produces colonies that have distinct green metallic stain. Most strains of E. coli also demonstrate beta hemolysis on blood agar. The varying strains of E. coli have specific virulence factors that determine disease characteristics. In the case of urinary tract infections, P. fimbriae are the primary virulence factors due to their ability to adhere to uroepithelium. Normally, E. coli is a commensal bacterium in intestinal tract. However, it can spread from the perianal region to the periurethral region, causing UTI. These are the virulence factors of E. coli, which we have discussed and we will be discussing in many other videos. So P. fimbriae allows adhesion to the uroepithelium in case of UTI. Thank you for watching. Keep studying.